I think now, and I think we can get Terry involved in this conversation, we have a nice adjuvant trial that's starting. Is that NSABP B55?
It's NSABP B53, but it's a SWOG-led trial. It's SWOG 1207, and we co-conducted, if you like, with SWOG. And it's adjuvant everolimus with patients with high risk, non-negative and not positive breast cancer. It's not accruing as well as one would have expected. Part of it is the design, because it's for patients with 1 to 3 positive nodes that have high oncotype score. And also for post-neoadjuvant chemotherapy, they had to have 4 positive nodes. And there's an amendment now that's circulating in SWOG to actually loosen the eligibility criteria to like any node-positive patients, and after neoadjuvant chemotherapy, any node-positive patient. So hopefully that will help the trial accrue.
That's a great trial.
Part of it is also we as an SABP still are sort of married to the B47 trial, which competes with this trial. So we'll put a lot of patients on B47. So once B47 closes, I think that probably will be the only adjuvant trial in ER-positive breast cancer we have available.
Yeah,
and so that will
I mean, are you guys aware of that trial, of that SWOG study, using the everolimus in the adjuvant setting for high risk? Are you participating in it? No. Are you guys participating in it?
I think the trial is really interesting. It's just too bad that we can't select who's going to benefit. We tried so hard to find that out, but we haven't been able to. But I think there's great interest in the outcome of the trial.
Yeah, does anybody have any good biomarkers for response to everolimus that you know about? I think we've looked at them all, haven't we?
I think it's pathway. Now people are trying to look at it's the whole idea of a signature. You can't be looking at one thing, a PI3 kinase mutation, for example. You have to really look at a combination of factors and interrelated pathways. And that's complicated, but I think that's the wave of the future.
Yeah, no, I would agree. Okay. Before we go on, I want to talk, we kind of briefly talked about palbociclib and CDK4 inhibitors. And does anybody have any comments about that? I mean, we can maybe start on this side, What do you think?
So I think it's very exciting data. Again, palbociclib is the first in its class. Now there are a number of other drugs being formulated by other companies that are going to be looking at this. But the data came out of Richard Finn and Dennis Slayman's lab at UCLA, where the molecule was tested across a broad panel of breast cancer cell lines, 51 breast cancer cell lines, and it was the luminal types of breast cancer, ER-positive breast cancer, that seemed to derive or be most sensitive with growth inhibition to the CDK4/6 inhibitor. And so this led to a phase 1 study, and I have a patient who is enrolled in the phase 1 study who's now 5 years on palbociclib and letrozole, having never progressed. The main side effect of this drug is leukopenia and neutropenia, and it's dose limiting. We have to hold her dose every 4 weeks when she comes in to restart her dosing. We have to hold it for several days. But no infections, no fevers, no significant problems, and feeling quite well. And when you compare that to the toxicity of mTOR inhibitors, it's quite nice, actually. Phase 2 data was final data was presented at AACR this year, and I think a publication is being circulated right now that shows a substantial improvement in progression-free survival. No overall survival benefit. You wouldn't expect that in a smaller phase 2 study. So the phase 3 study completed enrollment over a month ago in the first-line setting, and I believe they just completed the second-line setting, Paloma 3 study.
Yesterday. Really? Paloma 3 yesterday?
Last night, close to accrual.
Yeah. So we're going to have some interesting data, and based on the exciting data showing a rather substantial improvement in progression-free survival, the FDA is, and I don't even know what this means yet completely. I don't know that any of us know, but granting it a breakthrough status.
Yeah, I don't know what does breakthrough status mean?
Hope could probably tell us.
I don't know that the FDA is completely clear on that either, but I do know that what they've said is they're going to pick out the drugs that have the most potential for success. And what that means is that they're going to stay in very close touch with the companies about the data as it's emerging. And when trials have completed accrual, go back to the data that led to the breakthrough status to consider whether or not that could be a mechanism for accelerated approval. And that's what they're doing with palbociclib, actually, is to review the phase 2 data to see if that could be a mechanism for accelerated approval while waiting for the phase 3 data from Paloma 2, which is the first-line trial. Of course, at the same time, there's the development of other CDK4/6 inhibitors, the LEE-011 from Novartis that's being studied in a very similar Paloma 2-like trial, Mona Lisa, but also in combination with an alpha-specific PI3 kinase inhibitor called BYL719, and that combination is still being studied in the early phase. But then there's a third PI3 kinase inhibitor from Lilly, which abemaciclib, something like that. I might be missing a syllable, but that drug actually was the third. That was the third drug in the whole course. But what's interesting about it is very different from the other two. It has single agent efficacy demonstrated in a phase 1 trial using the drug, unlike the other two agents, and it causes diarrhea more than bone marrow suppression, completely different. So it's being actually tested in a single-arm phase 2 trial with the potential to use that for registration. So, you know, agree completely with the toxicity assessment and, you know, seeing patients on it. We've accrued quite a number of patients to the Paloma trials, and it's been really fun studies.
The last question really about this is that they now, you know, they're actually the trial with palbociclib. There's going to be an expanded access study that's going to open fairly soon. I mean, are you guys? Would you be interested as a
absolutely,
doctor?
absolutely. And I think, and I really encourage all my colleagues and patients to go on trials, but expanded access, a little bit easier for a community oncologist to
really
involve patients. Yeah, absolutely.
Harder for us.
And there's also a global trial that we're going to participate in. I don't know if you guys select the Penelope trial, which will be post-neoadjuvant high risk.
Right, I think that's what
ER-positive
Penelope is.
patients.
We are going to participate. Are you going to participate in Penelope?
Yeah, so
That's a great trial. That's,
Through
again, a great trial
NSABP?
for ER-positive disease post-neoadjuvant.
You're participating through NSABP.
Yeah.
We'll
Yeah.
participate.
We are, the iSpy group is also participating. We're partnering with the NSABP on Penelope, which is this trial looking at high-risk ER-positive disease, post-neoadjuvant therapy. So you can't just have disease and be low risk. You have to have disease and be high risk by this.
High risk in that thing? What's high risk?
It's an algorithm based on
You explain the
from
algorithm.
MD Anderson, which has to do with clinical tumor size and nodal status and some of the
Proliferation.
other.
What's left over
So,
afterwards?
yeah, it's not
Okay.
only, you know, just disease, it's how much disease and
And then you get a
it's
year of palbociclib with your hormone therapy versus placebo. And it's actually the cool thing about that trial also is that NSABP is holding the IND in the United States. iSpy is working with the NSABP on that trial, but there are sites all over the world, including Asia.
The German group
and
is
it's
leading
great.
the
It's run
study.
by what is it?
GBG.
GBG Group
GBG.
and Gunter von Minkwitz.
I think that's great. I think that's maybe the template for us to be able to do trials, you know, globally this way going forward.
Absolutely.
You know, being able to have a U.S. IND holder be able to do this and take the risk globally. I think that's just tremendous. I hope that happens more often.
Yeah,
You know.
and an interesting point about palbociclib is that the preclinical data suggests it's incredibly active in HER2-positive breast cancer as well. And that hasn't been sort of seized yet by the companies because it's a crowded field. We just spent an hour talking about all the HER2-targeted agents.
Correct.
But there are subsets of HER2-positive disease that co-express ER where we don't get good pathologic responses. Where using an agent like this that is targeted at a mechanism that might be more active would make a lot of sense. And so I think it would be fun to generate clinical trials in this realm, potentially in the post-neoadjuvant or neoadjuvant setting.